Welcome everyone to our third COVID-19 webinar series. This is Wing, the Congress Director of the World Vaccine Congresses, and I'm delighted you can tune in today for this very exciting panel put together. I just want to take this opportunity to thank our webinar sponsor, TriLink Biotechnologies, for support and help in delivering today's webinar with these great vaccine experts. Just quickly before we start, I'd like to remind you that questions can be submitted through the control panel, and we'll do our very best to bring these up. As it is a panel rather than a presentation, as our last webinars have been, please feel free to submit these questions throughout the session today, which is also video recorded, so you will get a link to it tomorrow. So now I have the pleasure of introducing back our wonderful moderator, Dr. Philip Krause, the Deputy Director of the OVRR CBER, the FDA, and as his title suggests, he oversees activities associated with regulatory review products regulated by the FDA's Office of Vaccine Research and Review. He also contributes to global public health as an ad hoc advisor to WHO, and most recently, development of a research roadmap for vaccines against SARS-CoV-2. If you've been following our webinar series, you'll know that Phil has been a fantastic moderator, best placed to help navigate such a big topic in a time of this panel with our speakers today. But over to you, Phil, to introduce the session and panelists.
Thanks, Wing, and thanks for the very nice introduction. But today, really, the focus should be on our panelists, because what we have is an amazing panel that expands all of the expertise that is needed to really gain a comprehensive view of where we stand with COVID-19 vaccines. And this includes immunology, it includes virology, it includes vaccinology, and we also have representatives who are developing vaccines and have been involved in designing clinical trials for vaccines. And of course, all of these individuals have a breadth of experience that spans COVID-19 as well as many other different pathogens. And so what I'm going to do is jump straight into the discussion with this group. And as we go into the discussion, I'll say a few words introducing each of the speakers so that you know a little bit more about them. But to start off with, I would like to address a question to Anne Moscona, who's at Columbia University and is an expert on viral entry in virology for a number of different viruses, including COVID-19. And that really comes down to the fact that vaccines need to be able to interfere with viral cellular interactions in a way to protect people. And one of the critical interactions that almost all vaccines interfere with is entry into cells, and that's in fact the target of most neutralizing antibodies. So tell us a little bit about the virology of COVID-19, of SARS-CoV-2, and what it is that the virus needs to do to get into a cell, and how it is that a vaccine needs to, how it is that a vaccine would interfere with that. Anne.
Thank you very much, Bill. It's a pleasure to be here and talk about this topic. So SARS-CoV-2 coronaviruses are basically a packet of genetic information enclosed in an envelope that is similar to membranes inside the cell and derived from them. And inserted into that envelope are the glycoproteins that mediate entry and a number of other functions, specifically the spike or S, as well as the M protein that is really important for membrane formation, and the E protein, which is a really important glycoprotein, surface protein as well, which is an ion channel. Now, this the first step in viral infection by coronaviruses and SARS-CoV-2 is interaction between the spike protein, or S, and the cell surface. As you mentioned, the sort of key initial step in starting off infection. And as most people know by this point, the spike protein interacts with ACE2 on the target surface. Now, the spike protein then has to mediate fusion between the cell and the outside of the cell and the inside of the cell. And for coronaviruses, distinct from many other viruses, this can occur either directly at the cell surface when the spike protein mediates fusion of the viral membrane with the cell membrane, or it can occur inside the cell after uptake into an endosomal compartment. And so this spike protein is actually very versatile. And a really key aspect of the spike protein is that it exists initially on the surface of the virus in a state which is protected, which is called the prefusion state. And protected inside that conformation of the protein is this part part of the spike protein called the fusion peptide that is necessary for actually fusing the viral membrane with the cell membranes, whether it's directly at the surface or internal. In order for that fusion to occur, the spike protein has to undergo a conformational change through a transient intermediate and then into this final state where it can then insert into the target membrane, whether it's at the surface or inside, and then undergo a refolding step where that protein actually again alters its conformation in order to merge membranes of virus and cell. And so this protein actually undergoes several conformational changes and is present in several different states, all of which are essential for the function of the protein at getting the virus into the cell. It also undergoes, in order to go through those conformational states, undergoes several cleavage reactions requiring proteases that are also essential for this sort of dance that gets the virus into the cell. So I think this protein...
And let me stop you there then, because as we think about the spike protein and its interaction with the ACE2 cellular receptor, and you point out that it can be present in many different conformations, and of course the receptor binding domain itself of the spike protein is only a small portion of that protein. What if we're trying to develop a vaccine against SARS-CoV-2, how do you think the antigen should be presented? What is the best thing to show the immune system so that you can get a protective immune response that has the best chance of interfering with viral entry?
Well, this is critical, and we have precedent for that question in several other viruses that have been hard to develop vaccines against. It's really important. It seems to be, from what we know so far, really important to develop a vaccine against the stage of S before it undergoes its conformational changes. So in other words, capture it before it already begins the entry process, which is going to be the pre-fusion state. And however, there is also a transitional state at which the spike protein is beginning to unfold and beginning to undergo the steps that it needs to do to affect entry, and that could potentially also be another target. So it's not just either or, but it appears most likely that we need to direct immunity against the stage of the spike protein before it's already begun the cascade of getting the virus into the cell.
But can I ask a question there, which is that if you're, I mean, in some ways, does it matter? If
before you ask a question, Peter, let me just introduce you. Okay. Peter Hotez is, of course, a world-renowned expert in vaccinology who has great expertise in tropical medicine as well, and is a strong advocate in understanding vaccine safety issues, and is himself working on developing a SARS-CoV-2 vaccine that may be appropriate for use in developing countries. And so with that, I will let you ask Anne a question or jump in with your thinking about the appropriate conformation for the spike protein.
Oh, thank you, Dr. Krause. It was a very nice introduction. So what I was going to ask Anne is, you know, in some ways, does it matter? Because if you can show that whatever form of the spike protein you use, or even the receptor binding domain, if you're inducing high titers of virus neutralizing antibody, does it matter? Or is that not a good enough or an accurate readout to shape as a correlate of protection? I mean, Dan Baruch showed in the non-human primate model for COVID-19 that it seems to correlate pretty well with virus neutralizing antibodies, and certainly we showed that with SARS-1. So if you have the high neutralizing antibody titers, does it matter if it's pre-fusion, post-fusion, or a peptide or anything else? And is the proof in the pudding just the VNA, the virus neutralizing antibody?
I think you're absolutely right. Neutralizing antibodies that prevent infection are what we want to induce. And while we think that the most likely way to get those is to direct a response, whether it's an antiviral, as you mentioned, an antiviral peptide or an antibody against the pre-fusion stage of spike protein, in the case of a number of approaches, including antiviral peptides, we're going to be inducing a response against, or providing a response against the transitional state. So you're absolutely right. What's important is preventing entry. And if we do that, that's the goal.
So to build on that, obviously one of the ways in which the spike protein can be held in the pre-fusion state is by making some mutations and introducing a couple of prolines into it. It was nicely shown by the NIH Vaccine Research Center, where Gary Nabel used to work, although he's now the chief scientific officer at Sanofi. So I wanted to ask Gary as well as Hanneke Schuitemaker, who's really the expert on vaccine development at Janssen, which also has a candidate for SARS-CoV-2. For the vaccines that you're developing, how do you see the importance of the antigen that's presented? Are you doing something special to modify the spike protein in order to get the best possible immune responses?
Yeah, hi, thanks, Bill. Let me just make a couple of general comments, and then I'll say specifically what we're doing with the Sanofi candidates. You know, when you look at any virus, and I think to both Peter and Ann's comments, what we have to deal with is the target on the virus as nature evolved it. And in some viruses, the spike proteins are evolved to basically mask the critical vulnerable epitopes that are needed for viral entry. In other cases, they're relatively well exposed. You know, for example, in flu, they're relatively well exposed. In HIV, they're very, very hidden. The work around exposing hidden determinants really started when I was at the VRC in the HIV area, Peter Kwong and Barney Graham. And Peter and Barney kind of took that to the next level initially with RSV vaccines, where they were able to expose hidden determinants in the pre-fusion proteins by structure-based design. And then that work took them to MERS, where they and Andrew Ward found this 2P mutation that you just referred to. And the 2P mutation does appear to open up epitopes that might otherwise be hidden and present an immunogen to the immune system that may be better than what we use naturally or what we see naturally in the virus. So the short answer to your question is that I agree with Peter, you have to be empiric. Neutralization is a very good surrogate. It looks like the aggregate of data right now suggests that the 2P mutation is very good at inducing neutralizing antibodies, maybe better than the native S form. I would say that probably almost all vaccine programs in terms of, including our own, have looked at these alternative versions of the spike protein, and you pick the one that seems to be the best. So so far, I would say that the 2P looks across all platforms to be an attractive candidate and would be a good one to include in a variety of vaccines.
Hanneke, is there something you would like to say in addition to that from the Janssen perspective or from the virus vectored vaccine perspective?
Yes, so I totally agree with what Gary just said. We have also applied that 2P stabilizing mutation in our vector presented as protein construct. And it is indeed sort of prolonging the time of the protein to be in that configuration in its exposure to the immune system. So you get more potent immune responses, not because of an artificial structure, but because you keep it in that more open, more immunogenic phase a little bit longer. And I think there is, you really need, it's a delicate balance between stabilization, but also to keep it flexible enough to get these good antibody responses. And I think the double P or the 2P mutation is one approach, but both Jason McLellan and recently Hans Langendijk from Janssen Vaccines have published additional stabilizing mutations that, of course, need to prove whether they would result in better immunogens, but there are more ways to achieve that potentially better immunogenic state of the S protein.
Excellent. So, of course, we're talking about development of neutralizing antibodies, which is one of the mechanisms which vaccines use. And yet, there have, I think, if one reads the literature, have been some questions about the importance of humoral immunity relative to cellular immunity and other arms of the immune system in protecting against SARS-CoV-2, especially as we look at individuals who've recovered, some of whom do not have very high either binding or neutralizing antibody titers against the virus. And so we're very lucky to have with us Akiko Iwasaki, who's a Howard Hughes investigator at Yale Medical School, who spent a lot of time thinking about viral immunology, not just for SARS-CoV-2, but also for viruses. So Akiko, can you sort of run us through your thinking about the overall immunology and what it is you think a protective immune response against coronaviruses, in particular SARS-CoV-2, would look like?
Thank you, Philip, for the nice introduction. Yeah, I'm also very happy to be on this panel. So yes, I have been thinking a lot about a variety of different immune effector mechanisms that can protect a host against SARS-CoV-2. And as we discussed already, the neutralizing antibodies are sort of golden standard for protection, particularly because it can provide sterilizing immunity if it happens within the mucosal surface. But there are also non-neutralizing antibodies that are protective by engaging innate cell types like NK cells or macrophages in order to allow them to kill infected cells or engulf infected cells for destruction. And as you mentioned, Philip, there's also the T cell immunity arm, which hasn't been paid a lot of attention to when we discuss vaccine work, but it's a critical arm of the immune response where T cell immunity is really critical for even driving antibody response, as T helper cells are critical for developing potent plasma cell differentiation that can secrete neutralizing or non-neutralizing antibody. And as well as they themselves have their intrinsic capacity to detect infected cells and eliminate them from the body. So in particular, the cytotoxic T cells that can detect infected cells and eliminate them, as well as there are a whole variety of T helper cell types like the Th1 cells, which are really great at recognizing antigen presenting cells that are presenting these peptides that are driving these type of immune response that are needed for protection. So I think that we need to think beyond just neutralizing antibody when we talk about protection of the vaccinated host.
Yeah, excellent. So Akiko, what can you say about the types of vaccines that you think are likely to yield the breadth of immunity that you think might be most useful?
Right. So I think every vaccine strategy has, strengths and weaknesses. In order to, of course, if you develop a very high titer neutralizing antibody, you may not need all these other T cell mediated immunity. Of course, you need the T helper cell in order to get those neutralizing antibody. Classically, live attenuated viruses and so on have been able to elicit very strong T cell immunity as well as antibody immunity. But we'll just have to see what each of these vaccine approaches will yield with respect to both antibody and T cell mediated immunity. And just to add to this, having a local tissue resident memory T cell population in the lung, for instance, that can detect immediately the infected cells and eliminate them is another sort of area of focus that I think we need to pay attention to. Particularly for protection against reinfection or primary infection.
Sure. So Gary, Sanofi is, I think, looking at several different methods for developing vaccines, including, I know, using a protein subunit strategies that you successfully employed against influenza. Tell us a little bit about the strategies you're using and why it is you've chosen the complement of strategies that you have.
Yeah, thanks, Phil. Maybe again, I'll start with the big picture, which is that when you're dealing with an emerging pathogen like SARS-CoV-2, there are many twists and turns in the road to a successful vaccine. And what you want to do is you basically want to have many shots on goal. And in addition to having the shots on goal, you don't want to be taking the shots from the same point in the ice rink if it's a hockey game, right? You want to be taking shots from different places, different strategies, passing off, that sort of thing. So we feel that it's prudent to look at alternative strategies. When you look across the field, there's really only one vaccine approach that has been used in a vaccine that's come to licensure previously, and that's the protein, the stabilized trimeric protein approach. That's one of our lead candidates. It's based on our flu block protein, which is a trimeric form of the flu vaccine that has gone to licensure and is available to a certain, and it's actually recommended in older populations for flu vaccination. So that's a tried and true approach. We have scalable technology, we have infrastructure in place. And from the SARS-1 outbreak, there was even a prototype that was developed. So the first approach involves trimeric protein. We will use that either by itself or with different adjuvants, and we're collaborating with GSK to combine it with one of its adjuvants that's been highly successful also in commercial vaccines, AS03. Alternatively, as Akiko mentioned, the presentation of antigen by these different vectors can vary. And so when an immune cell takes up proteins and presents it to the immune system by cross presentation, that's a different mechanism of presentation to the immune system than if you synthesize proteins endogenously within the cell, and then the cell can take up those peptides and present them to the immune system. That's what mRNA vaccines do. You saw probably last night that Moderna released its phase 1 results on their mRNA vaccine. It looks very exciting. We have an mRNA approach as well that we're pursuing with a collaborator called Translate, and so we feel that it's wise to have a little bit of a diversity in terms of the approaches that we will take. I should also mention, and I think, Phil, with your experience in vaccines, you'll appreciate this, there are issues that relate not only to the biology of the response, but also to the nature of the product itself. For example, a protein vaccine, we can store and we can ship without the need for cryopreservation. An mRNA vaccine, actually, at least in its present form, you need to have freezing, if not refrigeration, in order to distribute those vaccines. So I think that and then Hanneke will tell you about viral vaccines, which I think have their own advantages and limitations as well. So we're trying to create a bit of diversity, hedge some bets, have a few shots on goal, and most of all, play with this larger team approach to do our part to help respond to the virus.
Okay, well, Gary teed up this question for Hanneke very well. So tell us about the advantages and disadvantages of the approach you're using, assuming that your company will allow you to describe any disadvantages.
Only advantages, no. So we base our vaccine on the low seroprevalent At26 vector. So the virus is low seroprevalent, and we vectorized it to be able to deliver transgenes in the body that are then transcribed and produce the protein of choice. In this case, of course, the S protein. And to also link it to what Akiko said is the benefit of this platform is that it elicits both good neutralizing antibody titers, but also T cells. And I think especially in the discussion that has, of course, emerged also recently on durability of immune responses, I think the T cell support will be very critical and hopefully get durable immunity. Another advantage of the platform, and that's built from previous programs, is that we may be able to get already protective immunity after a single dose. And I think especially in pandemic situation that may be very helpful. A good aspect of the platform is that even single high dose that we see good tolerability and a good safety profile, which of course is also helpful in uptake, because if vaccine administration is associated with fevers or other unpleasant side effects, that may of course hamper uptake. But most importantly, we see very good immune responses across our programs. We see good neutralizing antibodies that are also durable. Of course, it remains to be seen what we will get from our COVID vaccine. We are not yet in the clinic. We have no data. We hope to start our phase 1 studies still this month. But everything looks on green light yet at this moment. And then finally, one more thing, if I may, is that we have good experience with the manufacturability, because that, of course, is also important, and we have a good stability profile. Like Gary said, we do not need -80 at the clinical trial sites, but we can work with refrigerated products in our supply chain.
And I think slightly contrary to what Gary said, your Ebola Ad26 vaccine did recently get licensed in the European Union. Is that correct? So congratulations for that.
Yes, I just moved from celebrating town hall to this webinar because we are, of course, celebrating a bit weird without being together. But indeed, we got approval for our Ad26 based Ebola vaccine that consists of an heterologous vaccination regimen with an Ad26 first dose and then followed by an MVA as a second dose. And there we, of course, through animal rule and immunobridging, we managed to establish a predicted efficacy signal based on which we received approval in Europe.
Of course, obviously, human clinical trials will be the acid test, which I think you'll have an opportunity to see with COVID-19 very soon. Peter, you also are working on a vaccine that you're hopeful will be useful in the developing world. Tell us a little bit about your strategy and what it is you hope to accomplish that's different from what other people are doing.
Oh, well, thanks, Phil. We've been working on coronavirus vaccines for the last decade. We partnered a decade ago with the New York Blood Center with Shibu Jiang, who's now at Fudan University in China, and Lan Yingdu, and they had been working for a long time on SARS-1 vaccines and showing that when they immunized animals with the full spike protein, they saw a fair amount of eosinophilic cellular infiltrates using the spike protein, and were concerned that this might be an indicator of immune enhancement. And they then moved to truncating that just with the receptor binding domain, were able to show that they could achieve neutralizing antibodies and protection without the immune enhancement, even with a receptor binding domain protein on alum. And we partnered with them. We're not really virologists. We develop parasitic disease vaccines, but we partnered with them and got support from NIAID and wound up manufacturing at Walter Reed a receptor binding domain protein that gave high levels of neutralizing antibody. And interestingly, on alum, it gave the least or almost really no immune enhancement, at least as measured by histopathology and eosinophilic infiltrates, whereas the other adjuvants actually did. So and then we actually looked at the literature and showed a lot of interesting literature showing that alum does reduce immune enhancement in several systems. So we thought that was interesting. Anyway, we had that manufactured, but we never then could get support later on to move it into clinical trials. And then COVID-19 came along, and we started making the COVID-19 receptor binding domain. And now we've done that. And it's quite interesting. All of the technology that we've used to make the RBD from SARS-1 seem to apply for SARS-2, including removal of certain amino acids for glycosylation and that sort of thing. And so now, and it looks like yields and production yields are really promising, and we're getting neutralizing antibodies. And the nice thing about making a recombinant protein in Pichia, which is the system we use in yeast, is that one, it's low cost because we can make lots of it. And second, that there's a lot of capacity in low and middle income countries for making pichia-based recombinant protein hepatitis B vaccine. So this is made in Brazil, it's made in Cuba, it's made in Indonesia, and India, in China, Bangladesh. And so that got us very excited about the possibility that perhaps we could develop this as a low-cost recombinant protein pichia vaccine for global health. And so my science partner for 20 years, Dr. Marilena Potazzi, and I got in touch with PATH based in Seattle, and they previously worked with the Gates Foundation to develop the malaria vaccine for Africa, the meningococcal A vaccine for Africa, and now we're pursuing this as a strategy to do scale-up production, and we hope to have some announcements soon, potentially for India and other countries. As I said, something that could be made locally by countries that have a lot of experience using yeast-derived hepatitis B vaccines.
Great, Peter. So you brought up, of course, a really important question with coronavirus vaccines, and that's this issue of immune enhancement. And of course, our understanding of immune enhancement goes back to, or our concerns at least go back to the 1960s when formalin inactivated respiratory syncytial virus vaccines were tested and given to children and actually made the disease worse when they ultimately got infected. And there have, in the meantime, for other coronavirus vaccines, been animal studies, at least for some vaccine candidates, that have suggested that that could happen also. And then, of course, there are different kinds of immune enhancements, some of which may be related to the neutrophilic and eosinophilic infiltration of the lungs, so-called enhanced respiratory disease that we saw with RSV. But then there's also a different phenomenon, which seems to be macrophage tropism dependent, called antibody dependent enhancement, which has also been observed with some coronavirus vaccines, including a cat coronavirus vaccine. And that also seemed to be antibody dependent. So let me start off with the immunologist in the group and ask Akiko, do you want to comment at all on immune enhancement and what you think are the major parameters that a vaccine, that one needs to consider in developing a vaccine to avoid a risk of immune enhancement?
And before Akiko, could I just say one thing nice about you, Phil, which is that you brought up one really important issue that I've been pushing very hard, and it's been hard to convince people, that the cellular infiltrates you see in the lung or the liver of vaccinated animals in challenge may not be the same phenomenon as antibody-dependent enhancement. And I think
you're absolutely right. They clearly are pathologically distinct. I would agree with that.
Yeah, but I think a lot of the confusion has arisen that people conflate the two. And
very good point.
It's out of guidance documents coming out of international organizations, and it's causing a lot of confusion, and actually, I think, leading to mistakes in prioritization of certain antigens or adjuvants, and so I'm really glad you just brought up that point. I'm sorry, Kiko, I just wanted to.
No problem at all. Yeah, so we've been thinking also about the antibody-dependent enhancement of disease and also infection. And so previous research that you just summarized, Philip, does show that this ADE can happen, especially when the antibody is not neutralizing or that they are developed to a low titer. And so in the setting of a sort of suboptimal immune response that's generated by either infection or vaccines, it is possible to develop antibodies that will not block viral replication, but instead, or maybe to some extent, but instead elicit immune activation either through Fc-dependent uptake into macrophages that either enhances entry and replication inside them, or they can introduce these endocytosed viral particle into the endosome, where toll-like receptors reside, and they can engage toll-like receptor signaling from within those macrophages that can elicit cytokine storm. And also, antibody at low level, even the same exact antibody at low level can elicit these kinds of responses from previous literature. So I think all vaccine development, I'm sure all of you in the panel are also thinking very closely about this, that we do need to pay close attention to the possibility of ADE. However, as I mentioned before, antibodies are not the only protective mechanism. So even if there was low antibody generated by a vaccine, if there is a robust T cell immunity that's associated, there's still the possibility of conferring protective immunity, even at the cost of a little bit of ADE.
So you had suggested earlier, Akiko, that non-neutralizing antibodies might also be protective, and certainly we have a precedent for that even with Ebola, where I was always struck by experiments that showed that monoclonals that were neutralizing sometimes didn't protect, and monoclonals that were not neutralizing sometimes did, suggesting that some kind of ADCC or other antibody-dependent mechanisms may also be playing a role there. And of course, that may be the case with coronavirus as well. And if we think about the very large size of the spike protein relative to many other proteins, and relative to the total size of the spike protein, the relatively small amount of that is devoted to the receptor binding domain. So what do you think about that in terms of developing neutralizing versus non-neutralizing antibodies and the risk, the potential benefit and risk of non-neutralizing antibodies in SARS-CoV-2 vaccines?
Right. So I think, in general, we cannot decide whether an antibody is beneficial or harmful until we have in vivo evidence. So there's a lot of in vitro assays that people like myself use in order to assess neutralizing versus non-neutralizing capacity of antibody. But in vivo, there are many, many different cell types that have different Fc receptors. Of course, the glycosylation of the antibody can also dictate which kind of Fc receptor it will engage, and that can determine whether it's going to lead to ADCC or some sort of beneficial antiviral effect versus those that would, promote entry into the cell and potentially stimulate toll-like receptors or enhancement of infection. So I think, unfortunately, right now, the real evidence will come from the in vivo studies.
Can I make a comment on that? So one of the things that I'm very intrigued about is the fact that in the rhesus macaque model, there's been now several published studies looking at different COVID-19 vaccines. And well, some of them are actually published, others are up on bioRxiv or medRxiv. And what's interesting, none of them seem to show any immune enhancement after challenge so far. So this includes DNA vaccines, adenovirus-based vaccines, even the Chinese Sinovac inactivated whole virus vaccine, which you think if anyone was going to show immune enhancement, it would be that one. So a whole inactivated virus vaccine on alum did not show any evidence of immune enhancement. And I guess the question is does that mean that we're going to be safe and out of the woods? And obviously we won't go to clinical trials, or is there something about the rhesus non-human primate model that they just don't get very sick, as some people have suggested?
I think that is an important end sentence, Peter, because I think the non-human primates are a very good infection model, but not necessarily a disease model. And maybe you can only pick up enhanced disease signals in a disease model, like, for instance, Syrian hamsters or maybe mink, which are not yet used, but we are, of course, facing problems with mink breeding and SARS-CoV-2 infections.
Gary, as a developer of a protein vaccine, you've obviously given this a lot of thought as well. And I suspect the adjuvant may also play a role here. But what are your thoughts on enhanced disease?
Well, I'd make two comments, Phil. One is, let's be careful not to use the word antibody-dependent enhancement too loosely. A lot of times we see the enhancement in cell culture assays, but how that translates to the biology of the infection in vivo is unclear. I'd prefer to call it vaccine-dependent enhancement, Because even for the well-known cases like RSV that you mentioned earlier, we still don't really understand the mechanistic basis of the enhancement. It's there, there's no question about it, but it's highly controversial whether it's antibody mediated or whether it's Th2 mediated. So I think we really need to be, just keep an open mind. What I would say is that I think the only answer will come from human trials, and that's why it's important to do the studies. I am encouraged by the comment that, by the observation that Peter commented on, that we aren't seeing enhancement in non-human primates. They have limitations, but that's a good preliminary signal that should give us some, comfort in going forward. So what I will say, though, and I think the key thing, and this is where I think as a field, I'm quite surprised that we don't know more about it. This typical course of the disease, where we see a mild infection, it disappears. Then in some people, it goes away, never to rear its ugly head again. But then those people who get severe disease, it happens at days 7 to 10, right when the adaptive immune response kicks in. So to me, that's kind of the critical unanswered question, both for disease pathogenesis and disease prevention. What's happening at that point in time? Are we seeing, is there viremia in the tissue that we're just not detecting because we're looking at it in the nose and the blood? Is there an adaptive immune response, whether it's antibody or T cells or Th1, Th2, that's just getting us off track? That's something we really got to get a handle on, regardless of what we do with the vaccines, because it's the thing that's causing mortality.
Well, and of course, Gary, as you know, the recent study showing that steroids can actually help people, at least with severe disease, suggests that at least some of the pathology is immune mediated, and so too much immunity might actually be a bad thing, depending on exactly how that is manifested.
Exactly, yeah.
So a number of people in the audience have asked questions about how we get appropriate duration of immunity to a vaccine, and they're discouraged by the fact that it may be that for coronaviruses themselves, the duration of immunity may not be that long. And that for SARS-CoV-2, there also are questions, at least based on some anecdotal data, of how long immunity might last. Of course, we have a record with many vaccines of being able to induce relatively long-lasting immunity. But of course, we also have a problem that a vaccine that we would like to get out reasonably quickly will be tested likely only over the relatively short term in humans. And of course, the immune responses, as we measure them typically in vaccine trials, are measured only soon after vaccination. So I would like to put this question out to all of you. What do you think about duration of immunity? What does a vaccine need to do to induce long-lived immunity, and hopefully longer-lived immunity than what we may be expecting to see with natural coronavirus infection?
Well, one to answer your question with a question that I have for Akiko and Gary is, we are, it looks like, seeing waning antibody titers with COVID-19. But my question is, does that really mean they are no longer immune? Are there still memory B cells and long-lasting plasma cells? So even though antibodies titers may be waning, and sometimes even to the point where they're undetectable, is it likely that there's still immunological memory there, and would that benefit in case they are exposed again to the virus? And what would that mean for boosting if they were to get vaccinated?
Well, on the other hand, Peter, it seems like people can get the common cold many times in their lives and probably do get reinfected with other human coronaviruses many times. And probably this is attributable to the ability of these viruses to evade various aspects of the immune system, whether it's innate, humoral, or cellular. So there may be a virological aspect to it as well, or not.
Yeah, if I could, add a comment here, I think what you both are circling around are the differences between the qualitative nature of the immune response and the quantitative nature of the immune response. So absolutely, Phil, there are times when viruses will evolve resistance by, mutation and, or in some cases, simply by the way their envelopes can adopt alternative conformations or glycosylation to mask epitopes. At the same time, it's quite common that you see this waning of the antibody response. And I think that it's when you start to see those responses drop down below a critical threshold that you are at some risk of enhancement. Dengue is a good example of where, particularly if you've had a mismatched dengue strain, you're at real risk of enhancement of disease. So I think the way I would look at it is, one of the best ways to, and by the way, Peter, I completely agree with you that memory B cells are there, and even at points where the antibody titers are low, they can come back quickly if the immunogen is the right immunogen, as to Phil's point. So what I personally would think about for some of these vaccines is, we need to get something out there to give people protection now. I think if we're smart, what we might start to do is take cohorts of those people and start to give them, some of them at least, vaccinations at later time points. My guess is if you did the mRNA vaccine at 0 and 1 month, and then came back and gave some of those people a boost at 6 months, my guess is you would induce some pretty durable immunity at that point. The same may apply to other vaccines as well. So I think that's an approach for future investigation that we can use.
Yeah, I agree. And I would like to add to the protection, the study that was already referred to by Dan Baruch, who had challenged non-human primates and then re-challenged the animals. And not only were the animals protected, but they also showed a very good anamnestic response. So even in an infection model with very self-limiting pathology, right? So they clear the virus very rapidly. That already elicited sufficient memory responses that could be recalled in that second challenge. So I do think that, to your point, although you don't see the antibody titers staying up for quite high, there is already after one limited infection already a memory B cell response build up. But maybe we should let Akiko respond, because the question was for you originally and we keep talking.
No, that's fine. Yeah, so I guess going back to the original question about the longevity of antibody response to coronavirus infection. So if you look at the literature from the common cold coronavirus human challenge experiments, what that shows is that within many of the primary infected individuals, the antibody titers did go down within a year or so. However, when they were re-challenged, while they were reinfectable, meaning that you can detect viral titer from the second challenge, most of these people were completely asymptomatic, meaning that in terms of disease protection, even without the detectable antibody, these people were protected. So there's definitely a non-antibody dependent immune response that were conferred in these challenged people.
Anne and Akiko, what can, obviously these are complicated viruses, and as we think about vaccines, we think about the spike protein, but the virus is doing a lot of other things, including immune evasion and so forth. What are other aspects of the virus that are important for us to think about as we develop vaccines?
I'm interested in, well, I just want to say that as a person who studies viral entry and thinks about antivirals, I'm interested in antivirals as a complement and to think about them simultaneously and synergistically with the development of vaccines. And in the regard of vaccines as well as antivirals, I'm interested in the E protein, which is a very important structural protein that is an ion channel and is involved in the budding process, assembly and budding, and prevents the spike protein from triggering into its fusion-ready state prematurely and permits budding out of the virus to generate hordes of new infectious virions that exit from the cell to infect other cells. And so I think that the E protein is potentially an interesting target that isn't as much discussed as the spike protein, but could be a very useful complement in our discussions. I think that this virus, in its life cycle, which is complicated, offers actually a number of interesting targets in addition to the spike protein, and the E protein ion channel would be one of them.
Mm-hmm.
Yeah, so from the immune evasion perspective, I fear that if the antibodies are developed in many people, and if there are escape mutants that emerge as a result of it, that's something that we do have to think about. There's no evidence for such mutants being circulating in human populations, but I do think that viruses are, selected for their ability to transmit from one person to another. And if they can escape a dominant antibody response that's generated by vaccine, it's just a remote possibility that we should consider.
We're down to our last 10 minutes, although we can go over a little bit. And so I wanted to ask you, given that this virus has now been around and available for study for, well, it's been around a little over 6 months and available for study for probably still under 6 months. What do you think in the last 6 months has been the most important advance? And what do you think is the most important thing, or what do you think is an important gap, something that needs to be done next? So let me start with you, Gary. Of what has been our greatest achievement so far, and what is the next important thing we have to figure out?
Well, I think, Phil, that to me the key opening move, like in a chess game, what you do in your opening move sets the stage for what you can do next. And I really have to applaud the folks who solved the structure of the S protein. I would say both the SARS-2 glycoprotein, and then I would also tip my hat to the groups that have been working on MERS and the groups that have worked on SARS-1. I think that we learned so much from those studies that we could apply immediately. So the genetics, the structure, and then this impressive movement, and I guess the only way I can refer to it is as a movement from industry to apply their various technologies to vaccine development. That combination is a really powerful one, and I think it does point to the importance of the long-term investments in vaccines, certainly from the governments and the NIH and the FDA and the CDC, but also the importance of the public-private partnerships. I think that we were able to move so quickly, and this really, when you think about it, is really astounding, right? There's not been an instance of a vaccine that's been developed in less than 20 years up to this point. We may well have one, within 2 years. So it's a testament to those investments, and let's hope that we continue those in a very responsible, and I would even advocate in a scaled up way. I don't think we're doing enough.
Are there achievements that panelists would like to highlight, or key gaps or places to go next?
No, I really would like to echo what Gary said, the public-private partnerships and also the responsibilities taken both by the private sector and governments, and especially want to emphasize the role of BARDA, who is really reaching out in an unprecedented way to not only give the funding, but also the scientific support and really, I think it's all coming together very nicely. I do think we have learned a lot from the response to Ebola, and also what was wrong at the time and what could be improved. And I think we now also see the blueprint at work that is showing already much improvement. But also to Gary's point, I think we can do even better and not be too afraid of working with industry. In this type of situation, I think there can be full transparency, and we can really work together without running with it, because I think we are all working towards the same goal, and that's making vaccine available to the world.
So, of course, one major achievement also, at least in my view, has been in a very short period of time, we've actually brought vaccine candidates forward that are already in efficacy trials. And so there's one efficacy trial that's begun and others that are soon to begin. And additional vaccines are to be tested soon. But it may be that we'll have answers, at least about whether some vaccines work or don't work fairly soon. And with that in mind, if you look at the WHO landscape, where there are at least 120 and perhaps more vaccines that have been announced, how difficult do you think it will be to actually come up with a vaccine that works? So let's imagine these 120 vaccines were all made it into phase 3 trials and all could be tested. Do you think that a high percentage of them will turn out to work, or do you think that the vaccine that actually works and gives us what we need will be more like the hen's tooth among those?
So
Yes.
I'll make a comment, which is I always like to refer. Carl Zimmer, the science writer, wrote an article, I think it was in the New York Times, where he referred to the COVID-19 virus, the SARS-2 virus, as clumsy prey, quote unquote. And in some ways, that's true. I mean, in many respects, this is an old school problem in virology. I mean, the virus is definitely doing things we haven't really seen before, with all the vascular events and the coagulopathies and things. But at the end of the day, it's a virus that docks with its receptor. And if you can induce lots of neutralizing antibody with some T cell responses against the spike protein, you'll have a vaccine. So I do think we will have lots of vaccines potentially that look very promising. I think the struggle is going to be making certain that we don't rush it and do something that winds up that's unsafe. And I actually feel pretty good that that's not going to happen, because, and I credit, Phil, your organization, CBER, which doesn't get the credit it deserves, the Center for Biologic Evaluation Research. We've worked with them for a decade. We consider them some of the most extraordinary vaccine scientists that we've ever worked for, and we learned so much from them. So I'm quite confident that they'll definitely hold the line to making certain that we don't rush anything, and really providing good regulatory science and oversight of what's happening. So I'm actually very optimistic that we will have multiple COVID-19 vaccines. The problem is going to be the pressures to roll them out prematurely, and that's going to be the struggle, I think. And so I've been saying, and that's why when I hear things, we're going to have a vaccine by the fall and this kind of stuff, I say, no, we're not. That's not the way it works, and we really need to do good phase 3 clinical trials confirming that the vaccines work and that they're safe.
Yeah, on that, if I can.
Go ahead.
No, I just wanted to say that, of course, if the first vaccine has an efficacy signal, then it's not so much if the others work or don't work, but it will be more difficult to demonstrate that they work, because, of course, you get into the discussion, can you still do placebo-controlled trials if there is already a vaccine available? Of course, supplies will be an important component then into those discussions on how you can subsequently progress with other vaccines that still need to demonstrate efficacy. And I think it will require some creative thinking on immunobridging or other type of studies that can be applied to get more candidates to the finish line, because it's not so much one winner, because I think supplies will also be critically important to get multiple vaccines over the finish line. And I think many of them may work, but if we get an opportunity to demonstrate that they work, is of course, well, that will probably require some more discussion, but give the word to Gary.
Well, actually, let me just, having heard that from you, Hanneke, point out that Hanneke is actually giving a talk tomorrow in this webinar tomorrow, also under the aegis of the World Vaccine Congress. And so you can hear more about the Janssen vaccine and how they've been working on their Ad26 vector vaccine tomorrow. Gary, what are your comments?
Well, I think there will be a window here to see whether more than one works, because one won't be widely available for comparison to the other as the first batch is coming through. So I think you'll see a handful being tested, and I don't think they'll all work. I think that some will work a little better than others, and I think more than one will work. The one other comment that I just like to leave with you is I remember when I first came to the VRC talking to Maurice Hilleman. Maurice was a mentor not only for many Merck vaccinologists, but to many academic vaccinologists. And I remember what he said to me at the time, which is, he launched a variety of pediatric vaccines that are still being used today. And what he said to me is, he says, every time I launch a vaccine, I hold my breath for the first 3 million doses. And that is a very thoughtful and wise comment.
COVID-19, that will be the first day probably, right, given the expected demand.
Absolutely. But it's that scaling and the long-term and the population effects where vaccines really declare whether they've got some hidden issue or maybe some hidden advantage. And so it is important to get multiple ones out there. I'm optimistic that we will. I am a little humbled by some of our, failures in the past. I think back to HIV and the pronouncements that were coming out of HHS when it was first discovered. So let's wait for the phase 3s and let's be optimistic. Let's push multiple candidates through, but let's also keep an eye on the big picture. That's all.
So I'm hearing cautious optimism that we'll get there. Although also I'm hearing that at least from members of this panel, it likely will take more than just a short time to get there. Is that a fair way to put this?
Yeah, but also, Phil, don't underestimate the anti-vaccine lobby is doing on COVID-19 vaccines. Right now, the surveys coming out of Reuters and the Associated Press will show that less than half of Americans may accept COVID-19 vaccines, even if they're available. And this self-inflicted wound, I think for all the good intentions of Operation Warp Speed, they've been horrible communicators. And actually, they've been that would be generous. They've been non-communicators and really tone deaf and oblivious to how aggressive and how effective in a nefarious way the anti-vaccine movement is. Even calling it Operation Warp Speed, I think, was a terrible mistake, because the truth is the clinical trials are not being rushed. It's it's accelerating manufacture and doing more shots on goal. So there's going to be an enormous amount of damage control that needs to be done, even if everything goes perfectly. And of course it won't. And every time something goes awry, that's really going to create a problem. So I've been pushing anyone who will listen to really shaping a communication strategy now around both the vaccine program in the US and internationally. And so far it's not really happened. And I know this because I go up against the anti-vaccine people a lot. I have a daughter with autism, wrote the book Vaccines Did Not Cause Rachel's Autism, so they target me a lot. So I know their techniques and what they do, but we have to be very mindful of that.
A very fair statement, Peter. And in that regard, I will point out that the FDA released a guidance document on vaccine development for COVID-19 that emphasized the importance of rigorous phase 3 studies with very rigorous success criteria as well, so that we don't end up with a vaccine that is weakly effective or not effective at all, and that we have adequate information on the safety of the vaccine, including the potential for inducing any kind of enhanced disease, whether it's antibody dependent or respiratory or other vaccine-associated problems. And I'll point out also that the WHO has been a real leader in this also, and at international fora has also really emphasized the importance of rigor in studying these vaccines and making sure that we don't make decisions that are too rapid. it's an unusual situation where many vaccines are being studied at the same time. And it's a statistical phenomenon that when you take lots of shots on goal, the chances that one of those shots will end up winning by a statistical fluke increase also. And so the world finds itself in an unusual situation, and this is not the time to relax criteria, absolutely. And I think that's a message that all of us can continue to try to get out there, that that it's so important to make sure that we know a lot about these vaccines before they're broadly distributed. Because as you say, Gary, the 3 million doses that Maurice Hilleman used to worry about being distributed over a year or two will probably be distributed in the first day. And we will see an unprecedented distribution of billions of doses of vaccine worldwide. And so really, we want to have a lot of confidence before those kinds of decisions are made with the lives of people around the globe, even if we think we're helping them. So are there any other comments that members of the panel would like to make in closing? We're a few minutes over, but I've really enjoyed this discussion with all of you. And I think we've certainly learned a lot about all of your perspectives. And my cautious optimism is reinforced by hearing what you all have to say.
Thank you, Phil. It's been a pleasure to join you today. Great panel.
Yeah, great panel. Thank you, Phil.
Well, and thank you all, and thank you, Wing. Wing, if you want to just make a brief announcement again about Hanneke's talk tomorrow, because I don't remember what time it is.
It's
that would be helpful.
Eastern Europe, yeah, EST time. So please join us back again, and I think you will have a presentation and be able to go much more in depth than what she has been able to do today. But again, you know, like echoing everyone, thank you so much for joining us. Thank you for all our speakers. There's been no lack of you know, interaction and good discussion, and likewise from our audience as well, for all your questions too. So thank you so much for your attention, and again to TriLink's Biotechnologies for their support, and hope to see you all again tomorrow.
Thank you.
Thank you again, and look forward to seeing all of the panelists in other fora in the near future.
Thank you, Phil.
Thank you. Goodbye.
Bye.
Bye.
Bye.